All right, now in 10 minutes, we'll go over the ion torrents. These are just some of the basic statistics. This is one of the ion torrent machines, PGM. You can basically decide which of their chips to use. Higher the chip number, the more expensive, and the more reads that it can sequence. Okay, so the ion torrent system. This is also sequencing by synthesis. Like Illumina, it will require an amplification step of your DNA, unlike PacBio, which just takes one of the original strands of DNA. It differs from both of them because it is non-optical. As you can tell by its name, what it does is it detects the hydrogen ions that are released during the polymerization of DNA. Instead of going through and attaching a fluorophore to all your nucleotides. And it also, and I'll get into why, like the PacBio system, tends to have some issues with homopolymer repeats. So if you have a bunch of A's in a row, it will cause the ion torrent a little bit of issues as well. So how does this actually work? Well, pretty much like everything else, first you have a special set of adapters you have to attach to your fragments of DNA. These fragments are then attached to this thing called an ion sphere. And what happens is each fragment is then PCR amplified in something called emulsion PCR. So each of these spheres will have many, many, many of the copies of the same piece of DNA. And then what happens is this solution is spread out on top of the ion sequencing chip. And here's a little bit more detail on the well. There are all these sort of micro wells. And then there are these other underlayers, which are actually done by semiconductor technologies that provide the detection steps for these. And here's just a little quick schematic of each one of these cells. They basically look the same. So this is what actually happens in each of the wells. And this is sort of what happens. This is similar in a sense to the PacBio system, in that you basically spread out your solution that's full of these ion spheres that have attached DNA to them, and they will randomly distribute into the wells. In the ideal case is one per well. This seems to do a lot better than the PacBio. The PacBio system is a lot more finicky. I should say that 150,000 wells, generally our best is to get half of them single occupancy. I believe with ion torrent, the typical industry number is 70, 75% single occupancy. So it's better in that respect. And basically what goes on here is this is a very sophisticated sort of voltage detector that detects when the pH of the well is changing in response to the synthesis of the DNA. Now, how is that actually working? So basically what you do is you basically have cycles in a sort of similar manner to the Illumina technology. But you do, and this is how it's different, is that you run each type of nucleotide one at a time. So you have a strand of DNA. You are waiting for, let's call blue T. Polymerase is waiting to incorporate a T, or I guess if blue is an A, then it's, or blue is T, then it's waiting to incorporate an A. So the system will flow through a bunch of A's. If A was not the thing that it needed, nothing would happen. If A is the nucleotide would happen, then we'd have what happens here. It gets incorporated, hydrogen ion comes off. There are enough of these strands attached to the ion sphere that produces enough of a signal that the chip will detect it. Now here in this third part is where we get into a homopolymer issue, which is if you're flowing through a particular nucleotide and you need more than one of them, it's going to release multiple hydrogen ions. Now, in some sort of very, very clean, non-stochastic world, this would be fine in that it would say, oh, I'm getting about twice as many hydrogen ions as I expected. I'm just going to call that, you know, a double G or whatever. And in fact, when it comes to just a couple of homopolymer repeats, the ion torrent system is more or less okay. It gets no problem when they start getting, you know, 4, 5, 6, 7, 8, that's the part where the fact that not all of those are going to have been incorporated in one cycle right away. So it's going to start messing up its signal quality. So in this case, in the PacBio's case, it's so long as there are more than 2 of the same nucleotides together, there's a potential problem. And that one, that you have 2 next to each other, or 3 next to each other, or 8 next to each other, is sort of not, it doesn't increase the complexity for PacBio. Whereas for ion torrent, the longer a homopolymer is, the more and more of a problem it becomes. That's how they sort of differ in their approach to or their sort of error profiles. And other than that, the sort of ion torrent basically works like the other things. It's just detecting the change in the pH as opposed to the release of a fluorophore. Do I have the recap slide? And one of their claims is that their technology should be getting cheaper and cheaper and cheaper as the sort of semiconductor processes that make it get cheaper and cheaper and cheaper. Because unlike a lot of other sort of biology-specific things that, it's only driven by the market for biotech, the semiconductor industry is being driven by the need to make smaller and smaller chips. So in theory, all the research that goes into that, as it trickles down to lower price points, should be able to make the ion torrent sort of a cheaper and cheaper method of doing the sequencing. So yeah, that's my quick overview of the ion torrent. And this is just a quick recap. Illumina sequencing, the best deal for your dollar. Like I said, it's the most mature technology. You will find many papers on it. You'll probably find something of whatever research, whatever project you're trying to do. You'll probably find a paper about it. Someone may have even written a program for you, because it seems quite often that people will constantly write a new specialized tool for very specifically what they're trying to do. They're also trying to get into sort of this more rapid sequencing. It's faster, but it's an increased cost. I should say that in terms of cost, Illumina, least expensive. Ion torrent, I guess, next. PacBio, probably the most expensive per base pair at the moment. But like I said, it's a very specialized sort of thing. So there are things that, there's no other way really right now. If you really need to get some 20K long reads, that's the only way you're going to get it, right? So there I feel the sort of cost per base pair equation doesn't really make as much sense to talk about, because it's there for very specific things. Yeah, and sort of strengths that they want to play up is the fact that you're actually just sequencing one piece of DNA. You have not gone through any PCR steps. It also has some other uses that I didn't really go into. For example, because you are using basically your original DNA that you isolated, they've been trying to sort of also get into this niche market of being able to detect things like DNA damage and various different types of methylation. So yeah, so PacBio sort of has its very specific purposes. And then ion torrent, the main thing I think is they want to push themselves as being like, sort of they have the least amount of sort of setup and time. In theory, the time between having your sample and getting its sequences should be the fastest, but it's still not quite as cheap. I also noticed when I was like looking over some of their marketing stuff, I think they've really been pushing themselves to be a lot more into people who want to do PCR products and amplicons. Like I said, they do not have the same sort of issues with, how complex is your sample variety within your sample. That's an issue for Illumina, not really an issue for ion torrent. And that is it. Any more general questions? None? Okay. So like I said, I believe on Thursday, I will have another class that's going to be an overview of how you actually analyze the data. It will, in fact, focus on Illumina data. So some of it could basically apply to ion torrent data. I will go over some basic things about you get your data, looking at your data, quality checking your data, the basic ideas behind mapping your data to the genome, and then I will go over briefly the sort of concepts behind the two main sorts of analysis, which were at the top of the slide. I talked about experiments, which is I will talk about ChIP-seq analysis and RNA-seq analysis. I'll probably give some examples, but it won't be an actual hands-on type thing. I'll mention this again then. What we've sort of been trying to do with some of this sort of the teaching aspect that we have of the bioinformatics facility is that we might actually move towards, like once you get the basic ideas of the analysis, we are thinking of moving more towards like small group tutorials. And often, even perhaps once people actually have their data sequenced, maybe even sort of take them through the analysis of their data. I've found that people working on their own data are a lot more motivated than working on some example data.